So thank you for having me here. It's wonderful to be back in the school. This building did not exist when I was here. I graduated in '95, so that's a long time ago. The high school corridors are the same, the entrance is the same, so it's nice to be back in a familiar environment. Today I thought I'd share with you the type of work I do, which is using human genetics to understand human health and disease. Basically, I really study children who come in with a life-threatening infection, and we propose the hypothesis that actually there's something wrong with the genetic components of the child. So this is the kind of work we do. But just to give you an overview of sort of how I got to where I am. So I was born here. My mom's Japanese and my dad's Spanish, but I was born and raised here. I was in St. Maur from Montessori to 12th grade. So I spent 15 years of my life here. So this is really home to me. The school feels like home to me. I did the IB diploma program. I took biology subsid, even though I had the option to do higher, but I was still able to make it through biology in university. So, so if you're not sure, don't worry about it. Things will work out. Then I next went to McGill. So I did my bachelor and my PhD training at McGill University. Does anyone know where McGill is? It's obviously in Canada. Nope, it's in Montreal, so it's on the east coast of Canada. It's a fantastic city to visit, and it's a fantastic city to be a university student in. So I know it's not part of I don't know how it is now, but when I was going through college selection, I got the impression that we were pushed towards the American universities a lot because we get a lot of exposure to them. But consider Canada, because actually it's much cheaper, the tuition too, but the level of education is fantastic as well. So consider those options as well. So I did my bachelor's in biology there, and then I went straight from a bachelor's into a PhD program. Usually you go through a master's program, but they had an accelerated program where I could do a 6 years PhD. So it was like doing 2 years of your master's with 4 years of your PhD. So that's what I did. And I did that in human genetics. And after that, so after we get a PhD, most people have to go on to do even more training, which is called a postdoctoral fellow. So I did my postdoctoral training in France. So I moved to France, in Paris, in the Necker Hospital, which is actually the first, it's where the world's first children's hospital was created. And this was created in 1788, a long time ago, actually 100 years before, even before the Pasteur Institute, which is also very well-renowned and is in Paris as well, was created. And so Necker Hospital is a wonderful place to do pediatric research, and that's where I did my human genetics and childhood disease research. It was a wonderful place to live. Paris is a wonderful city, and we had a wonderful view of Les Invalides and the Eiffel Tower from where we worked, so it was a very nice working environment. Once we finished my postdoc, so I spent 6 years there, then I was trying to find a real job. So finally, well, my husband also was doing his postdoc in Paris. We then moved to London in the UK, where I am now, and I've been there for 7 years now. And I moved to Imperial College London, which is one of the 3 colleges that are in London. So I think probably the well-known ones are Oxford and Cambridge, which are in Oxford and Cambridge. It's not at all in London. We do have 3 very, very good universities in the City of London: Imperial College, King's College, and UCL. So those 3, consider those as universities as well, because they have fantastic programs, and it's IB compatible as well. So I work at Imperial College, and I have a lab there now, and I'm located in St. Mary's Hospital. And St. Mary's Hospital is actually very well known. So it's been around for, I think, 150 years. It's a very old hospital, and it's in Paddington. And it's also the place where penicillin was discovered. So the actual lab where Alexander Fleming discovered penicillin is still there, and there's a little museum, and you can go visit it. And so it's a very sort of exciting historical artifact. The other more recent thing that's associated with St. Mary's that maybe you have seen in the news is it's where the royal babies were born, and mine. So the 3 royal babies were born in St. Mary's. The obstetrics center is literally across from my office. So when Princess Kate was pregnant, there was a huge crowd of mass media taking over the entire campus. So that's sort of our claim to fame, I guess. And we do actually quite exciting research there as well. So that's where I'm based. So just before I explain the kind of work and research that I actually do, I just wanted to refresh some refreshers on just basic concepts of human genetics. I'm not quite sure exactly what you've been exposed to. And maybe I didn't say this, but just jump in and interrupt if there's something that's not clear, or if you want me to elaborate further, okay? So I wanted to just reinforce the notion of genotype to phenotype. So this whole idea of inheritance. So your genotype will determine your outward expression of physical traits. That's the basic of what genetics is. And then obviously the variation in your genetics will determine your phenotypic expression. And how do we then determine genetic variation? We use sequencing technology, so I'll talk a little bit about that. And then finally, sequencing technology is absolutely necessary to carry out genetic studies. And so these are the few concepts I'll just sort of refresh for you so that at least the type of research I do, you can make sense of. So very, very simply, genotype to phenotype. So we have different alleles. So actually, we inherit 2 copies of genetic material. One full set from our father and another full set from our mother. So we have 2 variants or 2 alleles for every single gene. And those 2 alleles will determine your genotype and then hence your phenotype. So alleles are basically just different variants of the gene. So then the different alleles will make up your genotype. So you can have, say, an AA genotype if you inherited a capital A and a capital A from your mom and dad. You can have an AB genotype if you inherited A from your mom and B from your dad, and so on and so forth. And depending on those combinations, you will have a slightly different phenotypic expression of the gene. So you'll have slightly different physical characteristics. Next, so what are genetic variations? So these are just changes in your DNA. That's all it is. And very broadly, we can divide them up into 4 different categories. One are mutations, and these are usually 1 or more DNA base pairs that have undergone a change. And mutations, the other important thing is we call mutations things that are quite rare. And by rare, I mean that it occurs in less than 1% in the population. Then we have SNPs, or single nucleotide polymorphisms, which essentially is like a mutation, except the big difference is that is how frequent they are. So SNPs are frequent. They're commonly found, and so we define them as occurring in more than 1% in the population. So mutations are rare, SNPs are not. We can also have larger scale rearrangements, so not just having 1 base pair change or 2 base pair changes. We can have deletions of an entire region of a chromosome. We can have microdeletions of, say, 100 base pairs, or we can have insertions or genetic rearrangements like chromosomal translocation. So you can have an arm of the chromosome that sort of falls off and gets stuck onto the end of another chromosome. So these can also obviously affect the expression and function of those genes. And lastly, there's also, this is more a new type of variation that we've recently learned about. and that exists is copy number variations. So not only can you have actual changes in the makeup of your DNA, so in the base pair alignment, or you can have sort of deletions and so on, but you can actually have a difference in the number of copies of a gene that you have. So you can have, usually we have 1 copy of a gene or 2 copies of the gene, mom and dad. But sometimes you will inherit, say, 10 copies of that gene, and that will actually change the expression and the function of that particular gene. So copy number variants are also another type of variation that we have to take into consideration. And this is very new, and it's also very difficult to determine now. Now with the technology, we can actually detect it better, but it's still quite a new way of defining variation. So the other thing is, so we are, all of us in here share about 99.9% of our DNA. So if we sequenced all of us, we would come up as a 99.9% match. But that 0.1% of difference, obviously, if you just look around the room, makes a huge difference because we all look very different and act very different and so on and so forth. So that 0.1% of difference is extremely important and tells us a lot about the defining characteristics of each individual. Okay? And so of that, so for example, between you and me, on average, we would have about 1,000 variants, so single base pairs, that would be completely different and extremely rare. Okay, and something that we've never seen before. So you're walking around with 1,000 base pairs of mutations. You have 1,000 mutations. That sounds scary, but it's not, because we're all fine, which means those mutations probably don't have any effect. Okay, you can have just random errors. For example, if you're typing, you're going to make errors, and sometimes they'll make big changes into the meaning of the words. Sometimes it won't. If you put an extra space, no one's going to realize that you actually put an extra space there. So it's kind of that concept. In most cases, these variants have no effect on the actual gene. And then sometimes, so these are very, in the rare occasion, those changes will have a detrimental effect. Okay, so you can have one mutation that will just abrogate the function of the gene completely. And so this is, for example, with cystic fibrosis or sickle cell anemia, you can have a single mutation in the gene that will have a profound impact on your health or on the expression of that particular gene. On the other hand, you can also, just as rare, or probably even more rare, is that you can have a single change that will actually make the gene function better and will have an advantageous effect. It's just to identify that these genetic variations occur in different forms. Most of them will have no impact, and very rarely will actually have some change in the phenotype. Is that clear? Yeah. So how do we determine genetic variation? So how we determine it now, so there are many ways, and I'm only going to focus on sequencing because that's really the future of genetics, especially now that the sequencing technology has become very accessible and cheap. So pre-1990s and 2000s, so the time I was doing my work in the lab, we didn't have access to sequencers. And so the way we would do this was very laborious. We would run these massive sequencing gels, which are made from a very thin gel. And you would have to run these reactions that had radioactivity in it. So there was always this danger of, whenever you try to avoid using radioactivity at all in the lab, because there's always this, associated risks. But we had to do that for this particular experiment, and then we would run the sequence. And what's actually quite nice, and I found kind of beautiful, was that with these gels you would get these banding patterns. And basically what we have is in the first, you would always run a sequence of a fragment of a gene, and you would run it in 4 reactions. One that was the A base pair, the adenosine A, so hold on, I don't know if you can see that, A, C, G, and T. And the way we would do the sequencing is we would just read off the gel as we go down. So we would say that's C, G, T, A, G, G, A, and so on. And that's how we would actually read the sequences. And you can see it's actually sometimes very obvious if you see a mutation. You can see here, these 3 different individuals have a T at the end, whereas here we have different base pairs, actually 2 different base pairs. So this is just to show you how it used to be. And there's I don't know, artistic beauty in doing this, but it's very laborious, and the best you could do was read 600 base pairs at a time. So then now we have, since the 1990s, we have now this di-terminator sequencing reactions and automated sequencers. Don't worry about the specifics of it. I actually probably can't tell you too much about that either. The only thing is that we don't have to worry about radioactivity anymore. We prepare a special mix of our amplified gene. We put in different primers, basically saying this is the boundaries of the genes that we want to sequence. And you stick it into a machine, and basically you get, the computer reads out the sequence for you. Okay, so in a sense, we've lost that touch of being able to read out your sequence, but it actually is much faster. And this is the reason why the Human Genome Project was able to happen, which was completed in 2003. So that's basically what the output is now. You have the sequence chromas, is what it's called. So in each line, you would have, that would be 1 individual, and the little waves are basically the reading of the base pair. And you'll just read across, and actually the computer will read it for you. So in once you'll have all the sequence done. Then finally now, we've moved further on from this, and now we're in the era of next generation sequencing, which is much faster basically. That's the biggest difference, it's much faster. Why is it much faster? It's because before we would have to take our gene and amplify it to be able to read the sequence. And usually that took a little bit more time. Whereas now we have the technology that can actually read a single molecule of DNA base by base. So that just makes things a lot faster. And basically also means you don't need much DNA to actually do the sequencing. So just to show you, really, the pre-90s was this massive gel, and actually there were so many errors with it because if you poured the gel wrong and you were transferring it, and if it fell, it would be a mountain of gel, and then you've lost hours of your work like this. So it could be a nightmare. And then, so the traditional sequencers that we use for the Human Genome Project were these type of DNA analyzers. So you would just put in your sample and it would read off the sequence using a laser. Now we've moved on to a handheld device, and actually there are now devices that you can plug into your iPhone and you can put a drop. So we're getting to the stage where you can actually put a drop of blood onto another attachment to the sequencer, which will isolate the DNA for you, and then will actually sequence in real time your DNA. So we're at the stage where we've moved from these massive instruments to something that you can use with your iPhone, okay? So this whole advance in genetic sequencing technology has really had an impact obviously with the Human Genome Project, but also in areas of health research as well. So most genetic studies are done in the research setting, and it's only now that genetic or gene sequencing is so much cheaper, has it sort of penetrated into the more the consumer-based population as well. So I'm going to touch on that. The kind of work I do is obviously research-based. We're very close to the patient, so it's linked with the clinical system in the UK. And so in terms of genetic studies, the classic sort of 2 types of population or individual-based genetic studies would be carried out using these genome-wide association studies. I'm not going to go into that because there's a whole theory behind it, and it's quite complicated, but the whole point is that it used to be, it only would pick up common variants. So remember I told you about mutations and SNPs. So these GWAS studies only focused on SNPs. So if you had a rare mutation, you would never find it using the GWAS study. So there was limitations to using this. The other limitation is that you need thousands of patients and thousands of controls to be able to carry out the study, okay? And so when you're talking about a very rare disease where we don't have 1,000 patients, this wasn't really a possibility. On the other hand, now with sequencing, we can actually sequence every single base pair. And especially with rare diseases, we can then focus on families, and we can actually carry out analysis and identify those rare mutations that can determine the risk or the reason why they're sick. So because I mentioned whole genome and whole exome sequencing, which is both types of next generation sequencing, do you guys know the difference between genome and exome? Nope. So genome obviously is sequencing every single base pair, whereas the exome is focusing on the 1% of our genome which has the protein coding genes, okay? So actually 99% of the genome is what they used to call junk DNA. It's not really junk DNA, it's just areas of the genome that does not encode protein. So we focus actually most of my work is focused around using whole exome sequencing because it's cheaper still. And number two, because actually for the types of diseases that I'm looking at, which is Mendelian disease, so single gene disorders that are very rare, it's actually usually found in protein coding genes. So we decided to focus on the exome instead of the genome. What we have here is the alignment of a sequence of an individual around a certain gene. And you can see here this is the whole exome. So you will see, this is the same area of the genome, but there's only one protein coding gene there. And so on the exome, you will only have alignments of the sequence of the individual around the protein coding gene. Whereas in a whole genome sequencing, you will see alignments of the sequence across the whole region, okay? So it's very obvious the difference between genome and exome. Okay, so just to take a step back, what could you learn from genetic testing or genetic studies? So obviously in the research setting, we were always interested in genetic testing and genetic studies. Basically, there are many reasons why you want to do genetic studies in a research setting, but they all basically come down to identifying functions of a particular gene. So, you know, if you're interested in a particular gene, you would then carry out various experiments around that. What's new with the sequencing technology becoming more available is the fact that it's now actually becoming part of our clinical workup, and it's being it's present in the medical setting as well as in the consumer-based setting. Okay, so in the medical setting, it can be used to assess disease risk. So if you come in with a certain illness and the doctor says, well, it could very well be that this is a genetic disease, they can carry out the testing. It's also, by doing this, we can also either tailor specific treatments or prevention to, if we know that you carry a genetic risk. And also we can tailor, so this is all the coined term personalized medicine is. So based on your DNA, we would tailor the treatment to you, okay? And the other way we can also, what we could learn from genetic testing is that you can test for your response to different drugs. We know that different variations and different genes can affect how well you metabolize certain medication. So, or your response to different types of medication. So that's also becoming sort of standard part of clinical care. Now the fun part, I'm not saying that's not fun, but from a consumer point of view, there's also this direct-to-consumer genetic testing that's now becoming available. I'm not quite sure in Japan if 23andMe has been here or has arrived. There are other ones, I think, that are around. So for that, what can we learn is your ancestry. So ancestry is kind of fun. You know, you think, oh, I know my ancestry, but once you actually do the genetic testing, you'll find some interesting things. And you get sort of a list of fun fact, physical trait outputs too, which, you know, there's no harm in it. It's interesting. You know, you have this hair color, this eye color, which may be obvious, but some other things like your lactose intolerance or things like this. Implications and considerations about genetic testing becoming more prevalent in the society. So obviously it gives us insights about our health, our behavior, our family history, and other traits. And it's highly personal information which may have personal and social and familial impact. So if you find out that you carry, or you're a carrier of a gene, say Parkinson's, and you've never seen it in your family before, all of a sudden now that information means that the rest of your family may be at risk, right? So what do you do in that situation? Or maybe you're okay with it, but your other, you know, and your brother's okay with it, but your parents don't want to know. So there are all these things, funny things that come out of the woodwork, and that needs to be considered. And social impact, health insurance companies may in the future not insure you because you carry a genetic mutation that predisposes you to, say, cardiac disease. That's not the case right now, but these are things to just sort of keep in mind. It's all new information, and you know, the government and the society is trying to understand how best to incorporate that into standard care. Obviously, it's the personal impact too. If you find out you're a carrier of a certain disease, mutation, obviously, you know, each person has a different response to that. So, okay, so just a quick note about 23andMe. I'm a geneticist, and we use controls in our lab. I can't do the type of genetic testing, well, I'm not ethically allowed to do it on myself. You're not supposed to work on your own samples. But I have used 23andMe, and many geneticists are very, very enthusiastic about 23andMe because they're very research friendly. Their outputs are scientifically sound. So I've done this in the past just to show you how it works. Basically, you order a kit online, you spit in a tube, it gets sent to the lab, and there's DNA. So actually, the genetic testing for 23andMe is not sequencing. They use a genotyping approach, which is slightly different. And then the customer then receives a result probably within 2 months. So what was cool for me, well, I know my dad's Spanish and my mom's Japanese, that my ancestry report came back exactly as I would expect. I'm 50% Japanese, and it says here, I can't read here, but 49.5% European, and the majority of that is from the Iberian Peninsula, which is Spain. And you can see here they have these really nice interfaces that really explain things quite nicely. And every time I come back here and I go anywhere, everyone looks at me and says, oh, you know, because obviously I speak Japanese, nobody believes I'm Japanese, and for once now I can say, look, genetically I am 50% Japanese, and people can shut up. But so it's kind of fun to just see, and you'll see that obviously this is not, you're never going to get 50% Japanese, or, you know, because the Japanese population is a mix of other populations that came from, you know, that migrated in the past. So in my case, there is Korean ancestry involved in that. And in the Spanish side, you'll get much more mixing. So there's mixing between the Central and the Southern European countries, as well as the Middle East and North Africa. So this is very reflective of the natural histories of the populations there. So that's kind of fun. Well, actually, but at the same time, the caveat is, for some reason, I light up all of America, which shouldn't happen, and that's only because the Japanese signature is linked to the Native American signature, but the Native American signature is actually, they've got it the wrong way around. I mean, it's not that the Japanese were derived from the Native Americans, obviously. The Native Americans came from Central Asia, so this is a little bit misleading. And then for traits, again, this is probably too small to see there, I'm so sorry, but you'll get things like whether you have dimples, whether you have red hair, whether you have wet or dry earwax, lactose intolerant. Some of them are right, some of them are wrong, actually. And if you then look to see why it's wrong, it's because obviously most traits are not determined by a single gene. They're determined by multiple genes. So this will give you the most common genes that are associated with the phenotype. And if you happen to be a carrier of a rare mutation in that particular trait, they would be wrong too. So, there are things that may not match up, but most do, and it's actually kind of fun. And the beauty about this is they always update new traits. So every so often, you'll get an email saying, oh, we've added a few more traits, go on and look. They also do a health report, which you can choose not to see, and it's actually quite substantial. So that's just a fun thing. So now it's getting into the type of work I do. So hopefully in the next sort of 10, 15 minutes, I'll be able to just show you the type of work I do. So how many of you think that infectious disease outcome is controlled by genetics? Hands up. One? Really? Two? Yeah, so I think most people do think looking at genetics and infectious diseases is kind of a strange thing, because infectious disease is obviously because you were exposed to the pathogen, right? But in fact, actually, genetics is a huge component of that. It's your response to the pathogen. So of course you need to be exposed, but there is a response to that pathogen as well. So genetics does play a role. So obviously they say it's a battle of the two genomes. It's a battle of the host and the pathogen that will determine the infection outcome. Now, so we've used genetics to understand our immune response to human infectious disease from the time Pasteur was around. And there's two large approaches. We can either use an experimental approach, or we can use observations from natural infections. The experimental approach you can use an in vivo model, which is you can either try and create an animal model of that particular infectious disease, or you can use cell-based studies, okay? Obviously, there are pros and cons for using both. And we have this idea of using forward genetics or reverse genetics. Forward genetics meaning you can actually say if you have two mouse strains, one dies from infection, one doesn't. You can try and figure out, trace back from the phenotype what the gene is. Okay, this is what we do normally, but it's also very a laborious process. Reverse genetics is actually creating the mutation in the mouse and then infecting the mouse and saying what happens. Okay, so there's two different ways, either just looking at what's naturally there, or inducing the mutation and seeing the effect of it. In the natural human population, we obviously can't do reverse genetics. We can't create a mutation in a person and then infect them. So basically our studies are about the observations of infection in the population. So we can look at this at the population level, or we can look at this also at an individual familial level. So my work has really focused on the natural observation of these phenotypes. Okay, so if you take, all of us have encountered numerous pathogens to get to where you are now. Okay, during childhood, we have hundreds of infections. And these are all part of growing up and becoming a healthy adult. We train their immune response, the immune system, to actually interact with these pathogens. So there's probably about, I'd say about 20 childhood diseases that everybody would be exposed to, and some of them you would know, things like chickenpox, just because it left a mark, or you remember the event. But most of the other ones you'd never even remember. Okay, so if you take any of these childhood diseases, they fall into this infection disease severity spectrum. So in the population, most of us, and I'm going to be talking about herpes simplex virus 1. Okay, so herpes simplex is just it's a virus that all of us, actually 70% of us in the room are probably infected, and probably you don't know you've ever been infected. And this is the case with almost all our childhood diseases. And then in about 29% of us would have herpes labialis, which is cold sores. So this is what HSV1 is mostly known for, is having cold sores, okay? But only a small proportion of the population, so those are those in the dark blue, would have this. And it's not a life-threatening thing. It's kind of a nuisance, but we live with it. And then in the very, very, very rare extreme case, this is really in 1 to 2 in a million people, you would get life-threatening infection by this virus causing a brain infection, which is extremely lethal. Right now we have acyclovir, this wonderful antiviral that has reduced the mortality greatly. But even if the child gets this treatment, they usually suffer from neurological damage from the infection. So it's actually a very, very scary infection as a parent, and it's really quite horrible for the child. So I focus on that red individual, okay? I look at that individual and say, something went horribly wrong in that person. And so if we can understand what went wrong there, we may be able to understand the disease process better, and we can try and maybe prevent this from happening. So this is the type of work that I do. Just a quick thing on the genetic theory of infectious disease. So infectious diseases back at the turn of the century, no one knew what caused infections. It was the major killer. We didn't have antibiotics. There was no vaccine. So people died from infections. That was the #1 killer. Not cancer, not cardiac disease. It was infection. And Louis Pasteur was the first to propose, and he, through his investigations, actually, he did this really cool experiment with silkworms. Silkworms were really important at the time. He found that in certain factories, they would have these outbreaks of silkworms just dying. And he figured out this was due to a fungus, and he was able to show that there's this fungus that actually caused the infection, which killed the worm. Prior to that, people thought there was like an imbalance of fluids in the body that caused infection. Okay, so Louis Pasteur was the first to propose the idea that microbes caused infection. And at the same time, well, maybe a little bit later, the idea that genetics can play a role in that was also developed. And this is just to show you, so this is 2 pedigrees of 2 very famous scientists, Louis Pasteur on top, and Charles Darwin on the bottom. And what you'll see here is on the top, we have Louis Pasteur's family. He has 5 kids, of which 3 succumbed to fever, okay? Fever meaning obviously infection, and they all died in childhood. And Darwin's pedigree, we have, does anyone know what the double line there means? No? So a double line means it's a consanguineous marriage. So there were first cousins, first cousin marriage, which wasn't that uncommon at the time. And they had 10 children, of which 3 succumbed to fever, okay? So really, this reflects the times, you know, there was no antibiotics. And here is a setting where we can actually observe the genetic response to infection. So definitely genetics is involved in controlling infection. So classically, the idea of identifying genes that control your susceptibility to infection is known as primary immunodeficiency. So if you carry a gene, if you have a mutation in a gene that makes you susceptible to infection, it will be classified as an immunodeficiency. So these are inherited or genetic disorders that affect the immune system. And probably the ones you're most familiar with is the bubble boy mutation, which is the severe combined immune deficiency syndrome. It can be caused by a single mutation, 1 base pair that's wrong. And any of the, actually there's more than the adenosine deaminase gene, RAG1, RAG2 genes, there's many more. But just 1 amino acid wrong, base pair change would cause this really detrimental infection. And the SCID patients, the severe combined immune deficiency patients, now don't live in a bubble. We have come up with bone marrow transplant as an option for them to go forward. It's still a devastating disease, and these children still suffer quite heavily. So I don't work with those kind of patients. I've focused on different type of infection. And I'll be talking to you about that just shortly. So what happens in my lab? So in my lab, I have about 3 PhD students, 1 postdoc, and 1 technician who helps me, and I'm also there. I like to be at the bench as well. And so what I do is I'm interested in defining understanding what went wrong in that red individual, so that child who comes into the hospital with a horrible course of infection. So what we do is we get samples from the child. It's usually blood. Sometimes we get skin biopsy as well, because that gives us more cells that we can actually study. And then we do various different experiments using the blood cells or the skin cells to try and identify what went wrong. At the same time, we combine this information with the clinical outcome of the patient. And then we also do sequencing of the patient. So we try to put all components together to come up with a diagnosis. And so I'll talk to you specifically about our herpes simplex encephalitis patients. Very, very briefly, herpes simplex virus, I told you, is a virus that we're all exposed to. There's nothing to be terrified of because most of us have met it. It's probably in you and it's in us for life, and usually doesn't cause a problem at all. And like I said, in very rare occurrences, it can cause devastating illness. This is the spectrum of disease that HSV-1 can cause. The most common is labialis, or the cold sore, yeah? So this is probably quite, most people have been exposed to it. And these other, so you can get gingivitis, which is the infection of your gums. You can get gingivostomatitis, which is all the mucosal tissue in your mouth. You can get herpes whitlow, which is on your skin. And you can get eye infection as well, which is actually quite painful, called UV keratitis. But these are less common than the cold sore. And like I said, in 1 in a million cases, you'll get this encephalitis, which is devastating. And so what we decided to focus on was these patients and say, so actually 20 years ago when we started the study, no one knew why these children got sick. And the sort of common response was, it was bad luck. But there is no bad luck. That's not a good enough answer, right? So we also looked to see, obviously, I mentioned, you have to be exposed to the pathogen. So we wanted to know whether the pathogen carried any sort of virulent mutations, if maybe the virus itself was slightly different and more aggressive. We know that in these cases that's not the case. So we said, okay, well, let's propose the idea that actually the child may carry a mutation that predisposes to this severe infection. So we collected these patients, and like I said, they're 1 in a million, so it's very difficult to get many cases, but in collaboration with my previous supervisor in Paris, we have now over 300 patients. And what we do is, so the doctors will contact us and say, we have a patient who came in with herpes simplex encephalitis, and luckily now, most cases, they will survive. So what we ask for is once they're better, we'll ask for them to be included in our study to study the genetics of herpes simplex encephalitis. We ask the parents to agree. If they agree, then we take blood. And then in some cases, we also take a skin biopsy, which sounds scary, but actually it's not such an invasive procedure, especially for a child. And actually, these are also babies. We just take a 3-millimeter punch biopsy. So we numb the skin, and you just take a little punch biopsy, tiny, tiny little piece like this. And then we bring that into the lab. And then we carry out functional screens, which we then try and figure out what's going on. So patient arrives at the clinic, we get a call saying, okay, we have the blood for you. So we run over to the hospital, which is very conveniently just in front of the labs. We take the blood, bring it back into the lab, and we take the blood cells, or we take serum and plasma, which is the protein part of the blood. We freeze that because we may use that to test different protein levels in the future. The blood cells will isolate immediately, and we'll do either infection assays. So we'll take the patient cells and infect it with HSV-1, with the virus, in the lab to see what happens. And we also use the blood cells to extract DNA and RNA that we will use for sequencing later on. And then we can also use these cells to create induced pluripotent stem cells, or iPS cells. Have you heard of iPS cells before? Yeah? I mean, I think in Japan it's been in the news quite a bit. I mean, Yamanaka is the god of iPS who discovered it here. And so it's, we actually take patient cells and we can make them back. We can take the blood cell and then coax them back into a more pluripotent stem cell state, which means that basically we can take that cell and then differentiate it into any cell type thereafter. Right now, with just blood cells, we can only do work on blood cells. We can't turn the blood cell into a neuron. But by taking the blood cell and converting into an iPS cell, we can then later differentiate this iPS cell into a neuronal cell and then do different studies. Because we can't take neuronal cells from the patient, obviously. So skin biopsy, we take skin cells, do the same thing. We can also create iPS from skin cells. And the other thing we started doing now is taking hair follicles. These are children. Obviously, we don't want to do any invasive procedure, whether it's taking blood or taking a biopsy. So we've tried to come up with other ways to create cells. And actually, all we can do is we can pick 5 pieces of hair. And it's not just about hair falling off. We have to make sure that the hair bulb is there, that the follicle is there. We can freeze it. And then actually we grow those out and we get keratinocytes, which is a type of cell, epithelial cell. And from that we can create iPS cells too. So from a hair cell, we can create any cell type from the patient now. And it's non-invasive. So what kind of infections do we do on these patient cells? So now we want to know, how does the patient respond? So we first look at viral infections. So how do they respond to the virus? We can do microscopy experiments. We have HSV1 virus that's green, and we throw it on the cells. And you'll see up here, this is a patient cell line. This is this is just regular phase. So this is just looking down the microscope, a light microscope. Yeah. And then this is with green virus. So you'll see that the patient has tons of green virus compared to a healthy control that doesn't, okay? And then we also do plaque assays, which is a way to measure the amount of virus that comes out of the cell. And then at the same time, we look at the immune response. So how does the patient respond to infection? This is looking at the proteins that the patient produces. So upon infection, you'll see that these are just happy fibroblasts. So we have blue, that's the nucleus, and the green is the cytoplasm. It's actually a cytoplasmic protein. And upon infection, you get this really distinct pattern of recruitment in the cells. So this tells us that this individual has normal response in terms of recruiting these proteins to the cells. We do various cell viability assays. Do the patient cells die faster from infection than controls? We look for different protein markers, which we do with ELISA or Western blot. So we look at different levels of protein as well. So we did this with our patients, and we identified actually mutations in the toll-like receptor 3 pathway. So there's actually 6 genes in that pathway. And what they all share in common is that they can't produce this very important immune protective protein called interferon beta. So we've shown this in fibroblasts. I'll just go through this quickly. And so we were able to show the defect in the patient's fibroblast cells. And for a long time, because this started before iPS was introduced, we were never able to show that this is what was happening in the brain, which is where the problem was. And so finally now we've done the iPS-derived neuronal cells. So this wasn't work I did. It was the affiliated lab that's done this wonderful work. And so they created the neuronal cells from iPS cells from the patient. And what they show is that actually we see that the neuron, actually the patient who has this mutation is unable to control the infection with HSV1. So this white that you see here is all the virus in the neuronal cells of the patient. And in a healthy control, you don't see any because the healthy control is able to clear the infection. So actually it's a very complicated story because with the genes that we identified in these patients, patient has a problem, so has no TLR3 in every single cell of their body, but they only present with this brain infection. So it was always very difficult to try and understand what was happening. Why don't they get sick from other diseases? Because TLR3 is important for detecting other viruses too. And what we found is actually that there is tissue specificity. So TLR3, the gene, can be expressed mainly in the brain, in the CNS, and in the periphery and the fibroblasts. But in the blood, actually there are other genes that pick up the slack of the TLR3 gene. So this is why we get this very defined phenotype. Okay, so I'm just coming to the end here. So I just want to talk about pros and cons of being a scientist. So because I think it's a very particular field, and I think if you don't get exposed to it, you don't really know what you're getting into, and you don't know what you're missing out on. So first of all, very flexible working hours. Academia is very relaxed. It's not a patient, so we do patient work. So when we have patients in clinics, obviously we run by the times of the clinic. But there is no, there's no clock. So we don't clock in, clock out. That's a good and a bad thing, because actually most scientists tend to stay very long hours in the lab. But there's at least not fixed hours. There's plenty of opportunity to travel, and I think this is a huge plus that nobody knows about. So there's tons of conferences every year. You get invited to go to very new and exciting places that you never would go yourself. It's all covered and paid for, and you get to meet people from all over the world doing the type of research you do. So it's actually a fantastic, that's a huge plus for me. I love to travel, so that's great. The other thing is you're always learning new things, always. Everything is, you realize actually you know nothing. The whole approach to science is you're always finding new things, which is fantastic. And it's really wonderful when you realize you've made a new discovery. It's very fulfilling, and especially if it's a patient-based thing, you realize it can make a difference. Even if it's just to that family, it can make a huge difference. The cons, the work never ends. It's probably true about most jobs, but in this case, really, a project never really comes to an end. There's always things going on, and actually, it's really hard to shut off, because you don't clock out, and it's something that you're always worried about or thinking. There's an experiment running, you're always wondering, is it going to end on time? We have a long training period. I told you 6 years of PhD, you have your 4 years of bachelor's, 6 years, 2 years of master's, 4 years of PhD, and usually 4 to 6 years of a postdoc. So that's a long time before you can actually do your own work. There's not much money in science. That's a problem and a reality, and it's very competitive. But that said, it's a very exciting place to be, and if you were in London, you guys would be more than welcome to come by and observe what happens in the lab. So that's it.
Thanks for watching. Please subscribe to our channel for more IB Science videos.